Good afternoon, everyone. I would like to thank the organizer for providing me this opportunity to present my project. My research interest, which is also one of the major focus in our lab, is to explore novel radiosensitizing strategies for improving treatment efficacy of DMG. In this project, our strategy is to target mitochondria and hypoxia as a novel approach to radiosensitizing DMG tumors. As we all know, radiotherapy is the only standard treatment for DMG. As a palliative treatment, it can only slow the tumor growth in a short period. For most patients with DMG, tumor will come back within several months after radiotherapy is completed. This has been considered to be largely due to either intrinsic or acquired radioresistance. Hypoxia has been considered as one of the most common features in solid tumors. It is a physiologically low levels of oxygen tension which develops from uncontrollable cell proliferation and dysfunctional vascularization. Not only hypoxia is a change in tumor microenvironment, it also modifies tumor metabolism and blunts the efficacy of radiotherapy. DMG tumors have also been reported to show low blood perfusion and regions of necrosis in a functional MRI study. This indicates that this tumor grow in a low oxygen or hypoxia environment that supports activation of hypoxia-inducible factors. Briefly, radiation kills tumor cells by inducing DNA damage, and oxygen is the most important factor to stabilize the DNA damage, so that tumor cells cannot repair those DNA damage immediately, leading to cell death eventually. Under hypoxia, oxygen is significantly less and they are not able to stabilize the radiation-induced DNA damage. Therefore, tumor cells can repair those damage and survive following radiation therapy. On top of that, hypoxia also stabilizes the expression of hypoxia-inducible factor 1 or HIF1, which is a transcriptional factor regulating many key enzymes located in the glucose metabolism. It shifts glucose metabolism from mitochondrial respiration to glycolysis, induces angiogenesis, maintains an acidic microenvironment, and promotes cell proliferation. This metabolic consequence collectively reduces the efficacy of radiotherapy. To target hypoxia, there are generally two approaches. We could either increase the oxygen delivery to tumor cells or decrease the oxygen demand of tumor cells. There are many clinical studies trying to deliver more oxygen to tumors. However, as most of the solid tumors have abnormality in vasculature that restricts the oxygen delivery and perfusion, this approach has been proven ineffective. In contrast, reducing the oxygen demand of tumor cells has been predicted by the mathematical model to be more effective, and this has been confirmed by preclinical and clinical studies as a more feasible therapeutic strategy. To reduce oxygen demand of tumor cells, we decided to go for drug repurposing. Not only this would substantially reduce the cost, but also shorten the translational timelines. Patients with DMG currently have no other options except for radiotherapy, so they urgently need a new treatment that is more effective. The aim of our study is to rapidly develop a more effective treatment for these desperate patients and improve their overall survival. To get started, we first look at one of the mostly studied drugs, which is called metformin. Metformin is a very interesting drug that attracts a lot of attention in the area of drug repurposing. It is a first-line treatment for type 2 diabetes. Many preclinical and clinical studies have shown that metformin has benefit in cancer treatment and prevention. If we look at its mechanism of action, metformin is also a mitochondrial inhibitor that blocks the mitochondrial complex 1 within the electron transport chain. Therefore, metformin can also be used as a hypoxia modulator which reduces oxygen consumption rate and spares some more oxygen in tumor cells. There have been plenty of published work on metformin using this strategy. As an orally available drug, it also crossed the blood-brain barrier very well. We did an animal experiment using metformin and radiation on a DMG orthotopic model. The addition of metformin improved the survival on top of radiation alone. However, this is just a proof of principle experiment. As we can see, the increased survival is quite marginal, and none of the mouse with tumor was cured. This is largely because that metformin can only reduce 10 to 20% of the oxygen consumption rate in cancer cells at a clinically relevant dose. So we definitely need a more potent drug to improve this therapeutic strategy. If we look closer at the structure of metformin, we notice that metformin as a member of bigonide is a hydrophilic drug. It enters cells by using the cation selective transporters. There's another bigonide derivative called phenformin, which shares a lot of similarities with metformin regarding mode of action when used as the anti-diabetic drug. However, phenformin also has some unique features that metformin doesn't have. The most important feature is that phenformin is a lipophilic drug, which means it does not rely on those transporters to enter cells. This would allow much higher intracellular concentration as opposed to metformin. When used as the anti-diabetic drug, phenformin is 50 times more potent than metformin. In addition, because it is a lipophilic drug, it can cross blood-brain barrier even better than metformin. So theoretically, more phenformin would reach tumor cells behind the blood-brain barrier. We've also noticed that higher level of drug and a stronger efficacy are hand in hand with higher risk of side effect. Both metformin and phenformin would have risk of acidosis, in particular in patients with renal dysfunction. Although phenformin has been put off the market since the 1970s, there are still plenty of studies repurposing this drug as a cancer treatment. When measuring cell proliferation, phenformin is 40 to 50 times more potent than metformin to inhibit DMG proliferation. To inhibit oxygen consumption rate, phenformin is 100 times stronger than metformin. We further look at phenformin's activity on other mitochondrial functions, including maximal respiration, spare respiratory capacity, coupling efficiency, and ATP production. All these were reduced by phenformin. It also shifts ATP production from mitochondria to glycolysis at low dose and reduces the net ATP production at high dose. In line with clinical adverse effect, the reduced oxygen consumption is hand in hand with a slight increase in glycolysis measured by ECA or extracellular acidification rate. We further did a metabolomic study and found major products in TCE cycles such as succinate, succinate-CoA are decreased, as well as products in other metabolic pathways such as 1-carbon metabolism, glycolysis, and amino acid metabolism are increased correspondingly. To use phenformin as a radiosensitizing drug, we need to take some action on minimizing its major side effect. This led us to a second drug called dichloroacetate, or DCA. DCA is a pyruvate dehydrogenase kinase inhibitor that can be used for treatment or prevention of lactic acidosis. By inhibiting PDK, the PDH activity is restored. Thus, pyruvate will be converted to isoCoA that is further metabolized in TCA cycle. And this shunts the glucose metabolism from glycolysis to mitochondrial respiration. As we can see from literature, DCA was investigated back in 1980s in animal models and 1990s in clinical trials for the treatment of acidosis. It was also evaluated in children with congenital lactic acidosis, showing positive results. When we add DCA on top of phenformin, there is a significant reduction in phenformin-induced glycolysis, measured by ECA or extracellular acidification rate. Furthermore, the ATP production rate is significantly lower in combination arm compared to single drug treatment groups, indicating these cells are challenged by metabolic stress with very low energy supply. What we also found was an increase in cysteine level induced by the combination treatment. Cysteine is an intermediate and a supplier of several metabolic pathways. It actively contributes to cancer metabolic remodeling on 3 different levels. In its free form, it controls redox homeostasis as a component of the antioxidant glutathione. It is also a substrate for the production of hydrogen sulfide, which feeds the mitochondrial electron transport chain and mediates ATPase and glycolytic enzymes, thereby stimulating cellular bioenergetics. It is also a carbon source for biomass production and energy production. So, the increased cysteine levels indicate that the combination treatment not only disturbs the homeostasis of redox system, which triggered the antioxidant signaling, it also forces these cells to utilize other bioenergetic pathways as an energy compensation. We therefore further examined the combination of phenformin and DCA on proliferation of DMG cells. What we see here is a synergism from strong to very strong synergy across a panel of DMG cells assessed by the combination index. To understand what is happening behind, we checked transcriptome by RNA sequencing and validated by PCR array. What we see here is a group of increased gene expression in response to stressful growth arrest conditions and treatment with DNA-damaging drugs. There are also increased gene expression in apoptosis, cell cycle arrest, ER stress response, and mitochondrial dysfunction. In contrast, there is also a panel of genes with reduced expression, including genes responsible for major DNA repair pathway, including base excision repair, mismatch repair, non-homologous end joining, and homologous recombination. This transcriptomics change suggests that the combination should further enhance radiosensitivity of DMG cells, given radiation kills cancer cells by inducing DNA damage. To validate what we got from RNA-seq data, we probed some protein markers for DNA double-strand breaks with gamma-H2AX, DNA damage response with phospho-CHEK2, apoptosis with cleaved caspase-3, and anti-apoptosis marker MCL1. All these treatment-induced protein change are consistent with their transcriptome change. Flow cytometry analysis also demonstrated a higher level of apoptosis induced in combination. The hypoxia inhibition was also checked by a 3D neurosphere assay stained with hypoxia dye in green. The hypoxia inhibition is also supported by HIF-1 alpha inhibition, which further improved the radiosensitivity of DIPG cells or DMG cells validated by gold standard colony forming assays. As a further validation, we checked apoptosis and DNA damage marker using 2 independent approaches, Western blotting and confocal microscopy. Both demonstrated the highest level of cleaved caspase-3 and gamma-H2AX induced by the triple combination arm. We are currently doing some animal experiments, and that's also the reason why I'm not able to be there in person or give live talk via Zoom today. Although we have shown a lot of promising data from the combination of phenformin and DCA, there are also a few limitations to be considered. First of all, neither phenformin nor DCA is an FDA-approved drug. Although we managed to reduce phenformin side effect by adding DCA, using 2 drugs with radiation more or less complicates the clinical translation. So what we do need is to search for a more potent and less toxic FDA-approved drug. Based on these positive findings, we did an unbiased high-throughput screening which could potentially identify more drugs from FDA-approved list. We did this with a drug library consisting of almost 2,000 FDA-approved drugs. By defining over 30% inhibition of oxygen consumption rate as an arbitrary scale, we've identified 80 hits from 8 classes of drugs that can effectively reduce the oxygen consumption rate in DMG cells. These hits were further filtered against more criteria, including blood-brain barrier penetration, less toxicity, higher oxygen consumption inhibition, and potential for future clinical translation. The best candidate we've found here is called autovaquone. Autovaquone is an FDA-approved drug to treat pneumocystis pneumonia, and it is also used in combination with proguanil to treat malaria. It has good safety profile with minimal toxicity observed in clinic, and its bioavailability and blood-brain barrier permeability are both good. We've tested this drug on DMG cells, and it shows very promising mitochondrial inhibition, with effective dose starting at 2.5 micromolar. For its toxicity, we tested up to 10 micromolar for 3 days, and we did not see any cytostatic or cytotoxic effect. Here we demonstrated autovaquone decreases the oxygen consumption measured by seahorse assay, reduces the hypoxia measured by neurosphere assay, and also improved the radiosensitivity of DMG cells measured by colony forming assays. In conclusion, we have demonstrated that targeting mitochondria and hypoxia is an effective and promising approach to improve the radiosensitivity of DIPG models. An example here is to use bigonite. Although phenformin is superior to metformin, its toxicity needs to be managed by introducing a second drug, DCA, with the combination showing better radiosensitizing effect and less toxicity. However, the triple combination is more complicated than 2 treatment modalities, so it may potentially complicate future clinical trial for dosage and treatment schedule optimization. Our drug screening assay identified autovaquone as another promising FDA-approved drug to improve the radiosensitivity of DMG cells in vitro. I would like to take this opportunity to acknowledge all the patients and their family who donated their tumor samples to make our research possible. I'm also very grateful for my team members, our collaborators, and funding partners. Thank you for your attention.